Good evening, everyone, and welcome to the 2022 Mautner Memorial Lecture. My name is Tracy Johnson. I'm the Dean of Life Sciences, and I'm a professor in the Department of Molecular, Cell, and Developmental Biology. I'm really honored to get to lead this extraordinary division that's made up of 6 departmental units: ecology, evolutionary biology, psychology, integrated biology and physiology, microbiology, immunology, molecular genetics, and molecular cell and developmental biology, and Institute for Society and Genetics, and if you say it really fast, you win a prize. But what UCLA in general has accomplished over the last 100 or so years has been really remarkable. But what's happened in the life sciences, I think, is extraordinary. Everything from advances in molecular sciences to study of ecosystems, from brain physiology to psychology and everything in between. And there have been extraordinary breakthroughs that have started here at UCLA in the life sciences. So I'm really honored to have this opportunity to showcase some of the exceptional talent in our division by having the opportunity to welcome our distinguished guest. So I'd like to say a little bit about the Mautner family. So UCLA presents this memorial lecture series every 2 years, alternating between the physical sciences and the life sciences. And this year, the Division of Life Sciences has the honor of hosting the series. I'd like to acknowledge my colleague, Miguel Garcia Garibay, who is the Dean of Physical Sciences, for being a really terrific partner in the Mautner Lecture Series. So we are really pleased that Louis Perkins and Kevin Perkins from the Mautner family are here tonight, and they have been committed to really helping promote the excellence in the sciences here at UCLA. This series is part of UCLA's really deep commitment to excellence at every level. But one of the really critical pieces of this is we get to showcase something that sometimes gets overlooked. So while many of you know about the remarkable discoveries in the sciences that have happened here at UCLA, know about the scientists who've been at the forefront of discovery, one thing that sometimes gets overlooked is the people who are really often doing the hardest work are our graduate students. They're at the core of the breakthroughs, and alongside their faculty mentors, they design and perform experiments, analyze data, write papers, but they also mentor our students, our undergraduates, and then often serve as the teaching assistants in the classes that we teach. Literally, this university would not function without the amazing talents of graduate students. And so as a campus, we really want to think deeply about how we can continue to support graduate student success. And we're profoundly grateful to the Mautner family because they have shared a generous gift through a legacy to UCLA in this lecture series in their presentation of awards to graduate students, which we're going to hear about in just a moment. So I'd like to now introduce Kevin Perkins, who is a native of Northern California and is continuing his family's legacy of supporting the Mautner Lecture and graduate student fellowships. So we welcome Kevin to provide a little bit more insight into the motivation behind the lecture series and announce this year's recipients of the Mautner Graduate Award.
Thank you very much, Dean Johnson. So really honored to be here today. Came in from San Francisco and just wanted to talk for a second about my grandfather, Leonard Mautner. He was a very bright guy, orphan at a young age, and went to MIT and basically became a successful electrical engineer and businessman, and was involved in various ventures. Actually taught at UCLA Anderson School of Business briefly. And when he was with my grandma, he decided to start this lecture series as a way to he was really into the sciences and making sure that people had an opportunity, the general public had an opportunity to hear from distinguished scientists and people in the fields that he really appreciated. And so there's a public lecture and then a scientific lecture following. and it was a really a big part of his legacy to have this lecture. And one of his good friends, Richard Feynman, was the inaugural speaker, who was a friend of his from MIT. Actually, they went to MIT together, and they hung out, and so that's they actually did some drawing together and really close. Anyway, so they began in '83, and there's some great lectures, including Paul Berg, Ronald Hoffman, and just distinguished people in their field. As a kid, I've been going to these lectures and really enjoyed them a lot. So we're pleased to continue his legacy, particularly the Mautner Graduate Awards. Very quickly on my grandma, who passed away a few years ago. For her, it wasn't, they used to have elaborate things at Riviera Country Club, and she was always really focused on not spending too much money on the kind of events for the VIPs, but rather spending more of that money for graduate awards and scholarship. And today, we're really pleased to give 3 scholarships to some graduate students who are really excelling in their field, as Dean Johnson said, and are doing a lot of great work. Firstly, to Celine Sneddon, is a 4th-year PhD student in ecology and evolutionary biology. Her current work centers on understanding of zoonotic viruses like SARS, COVID-2 spread within and among tissues within the body, with the aim of shedding light on factors that mediate transmissibility and pathogenicity, and thus understanding key determinants of pandemic risk. So that's really a timely area of focus. First one. Secondly, Miriam de Siqueira is a 2nd-year PhD student in molecular, cellular, and integrative physiology. Her research studies how adipose tissue architecture and metabolic adaptations change in response to obesity and interventions that impose metabolic function of adipose tissue. I don't even know what adipose tissue is, but it sounds very, very fascinating. Her research examines that obesity may lead to translational defects that promote proteotoxic stress. So congratulations. And lastly, Peter Back is a 5th-year PhD student in molecular biology. His research focuses on molecular mechanisms that allow widespread parasite Toxoplasma gondii to cause devastating disease in immunocompromised patients and infected neonatal stuff. Toxoplasma serves as a model system for the malaria parasite. His research intends to make contributions that enable the field to combat these infectious pathogens. So, again, congratulations. And as Dean said, your work's a phenomenal representation of UCLA, and we were really grateful, so thank you.
So if you wouldn't mind, if the 3 students, if you could just stand so that we can all see you and give you a round of applause and have a photo taken as well. We're so proud. Thank you so much. And thank you, Kevin, and thank you to your family for their generosity and their endless support of the work that happens here at UCLA. So now I'd like to introduce Professor Oliver Progoso, who's an assistant professor in microbiology, immunology, and molecular genetics, who's going to introduce our distinguished lecture. Dr. Progoso is an outstanding researcher and educator who studies viruses such as HIV. And these viruses, which are obligate parasites, use hosts that they infect in order to facilitate their life cycle, all while avoiding the antiviral defenses of the cell, of their host. The Progoso lab wants to understand how this happens, how that came to be, and what we can do to get ahead of viruses. So they take a really exciting combined approach that's interdisciplinary to study the interactions between hosts and their pathogens. They use a combination of molecular virology, biochemistry, evolutionary biology, and through these combined tools, understand the function and evolutionary framework of viruses. His lab has been able to uncover important virus-host interactions, and as HIV represents one of the major burdens to human health, particularly in underserved communities, Dr. Progoso and his lab hope that their research will contribute to making inroads into the global fight against this virus. More generally, what they've learned and what we continue to learn about viruses certainly influences how we think about really fundamental mechanisms in molecular biology. And I will also say that Dr. Progoso is an extraordinary teacher and colleague, and so it's really a pleasure to get to introduce him, for him to introduce our guest speaker.
Thank you, Dean Johnson, for the kind introduction. I'm really excited to welcome you all here this evening and to welcome Dr. Joe Darisi as the 19th Distinguished Mautner Lecturer. Dr. Darisi joins an elite group of scientists featured as previous Mautner lecturers. These include Nobel laureates in chemistry, such as Dr. Paul Berg, world-renowned plasma physicist, Dr. Roald Sagdiv, as well as a Nobel laureate and former director of the National Cancer Institute, Dr. Harold Varmus. Dr. Darisi is currently professor of biochemistry and biophysics at UCSF, as well as president of the Chan Zuckerberg Biohub, which is a nonprofit medical research organization that is affiliated with UCSF, UC Berkeley, and Stanford University. Dr. Darisi's interdisciplinary research combines genomics, bioinformatics, biochemistry, and bioengineering to study parasitic and viral infectious diseases in, importantly, a really wide range of organisms. The goals of his work are to discover and study novel and unrecognized potential biothreats, to develop faster, better therapeutic options for known as well as emerging pathogens, to use data-driven diagnostics for infectious diseases, and to importantly make these technologies accessible and freely available to the broader scientific community. This work includes really important research in the identification of, for example, the original SARS virus back in 2003, studies on Plasmodium falciparum, the causative agent of the most deadly form of human malaria, as well as pioneering technologies such as DNA microarrays and whole genome expression profiling. More recently, they've turned their studies to focus on SARS-CoV-2 and COVID-19, particularly through the Chan Zuckerberg Biohub, where they've really been instrumental in California's abilities to sequence viral SARS-CoV-2, which has been essential for epidemiological studies, as well as for PCR tests for better health throughout the state of California. Dr. Darisi is nationally recognized for his efforts. He's a member of the National Academy of Sciences, the National Academy of Medicine, and the National Academy of Arts and Sciences, of which I did not know those were different things. But I will say that in addition to kind of all these accolades that have been given to Dr. Darisi and that he's known for me, it's really about his ability to transmit his excitement and passion for science to the broader audience. And this is both the scientific audience as well as the non-scientific audience. So I first met Dr. Darisi back in 2003 at a conference called SACNAS, or the Society for the Advancement of Chicanos and Native Americans in STEM. I was just beginning my scientific career, and I, to this day, remember that conversation with Dr. Darisi and how he inspired me and how excited he made me for the science that I was about to partake in. So today, Dr. Darisi will talk about pandemic stories, science and public health in the era of COVID. So please join me in welcoming Dr. Darisi to the stage.
So first of all, I'd like to thank the Mautner family, the Perkins family, Dean Johnson and Dean Johnson's colleagues for extending this invitation to me to provide this lecture to you today. This is a great privilege. It's tremendous to be here. I'm really happy to see old friends and make connections. Oliver and Peter and all these wonderful people I've met today, and of course, Dean Johnson. I'd really like to tell you a little bit about what we've been doing the last 2 years. It hasn't all been that much fun, but I think it's important to look back and see what's happened and to give you a little taste for our experience. And I know that everybody's experience is different and unique, and UCLA had its own experience. But I'll give you a little insight into what happened in San Francisco and how we got there. But the first thing that I need to do is tell you a little bit about the Chan Zuckerberg Biohub. Many of you know what UCSF is, but not many people know what that entity is, the Biohub. And it will segue into how we got into the pandemic and where it took us. So bear with me while I give you a little bit of background on that institute, and then we'll segue into the rest of the talk. So what is the Biohub? First of all, where is the Biohub? The Biohub is not that giant stadium over there. That's the Warriors basketball arena. I wish that was the Biohub. The Biohub is just this floor of this building right here. So it's a lot smaller. We don't have a basketball team. But UCSF itself is sort of the upper left-hand side of the screen, and the bay is towards the right. And so we're kitty corner to the UCSF Mission Bay campus. We're a nonprofit 501c3 medical research organization. We're affiliated, tied at the hip through a master collaboration agreement with UC Berkeley, UCSF, and Stanford University. And the whole idea behind the Biohub was really to bring these three institutions together with the hub at the center to promote collaborations and projects that otherwise would not have happened. So right now, where is a bit of a data picture? We're about 138 employees at the Biohub at all ranges of the different scientific disciplines, from technicians all the way up to PIs and research associates and so on. We have a lot of different research agendas, which I'll touch on briefly. We have a great scientific advisory board. I'm not going to go through all of them, but many of these people are well known to the research community here at UCLA and are renowned scientists in their own right. Now, there's currently only one Biohub, the one at San Francisco, but that is actually soon to change. And I think it's important to get this little announcement out there that the Biohubs are going to expand. So these are additional research facilities that will be founded or placed in other cities to promote interactions among academic institutions like it did in San Francisco, although every Biohub is likely to have its own custom flavor and research focus. And so that's being led by my former co-president, Steve Quake, who's now president of the CZ Biohub Network, and Amy Herr, the chief technology officer at the network, who's also a professor at UC Berkeley and an engineer by training. So this is very exciting. There's one Biohub now, but there are likely to be more Biohubs. There will be more Biohubs in the coming years. All right, so what does the Biohubs work? The Biohub was really divided into three major areas. It's people, programs, and platforms. And I'd like to take you through each of these three and give you a little taste for what's going on, and then get to our research. So the people part is investigators. We really believe in providing unrestricted funding to scientists with great potential to do their most bold and risky work without the sort of restrictions that come with certain federal grant programs and other things, that is independent of RFAs or requests for proposals. Really, people who have great ideas should be able to put money to work in new and innovative ways, even when there's no preliminary data. So like the Howard Hughes Medical Program or the Packard Fellows, this program takes a little bit of a flavor from that and supports people to do unfettered work. And to date, we've spent over $200 million into the universities to support scientists to do what they think is great. This is the 2022 Investigator Cohort. It's actually kind of a mix of people, and they're not all there, but we just recently took this picture. I thought I'd show it. So it's a 5-year appointment, and they get basically $1 million to do whatever they want in the scientific realm. I'm not going to go buy a boat with it or anything, but if it promotes your science and makes your science better, you should be able to spend that money on that sort of things. And we think it's a really successful program. The other idea is that we want to promote collaborations amongst these three institutions. These come from our three local institutions. And so this is a little graphic diagram of exactly that. These little arcs that go between Stanford, Berkeley, UCSF, and the Biohub show collaborative projects and papers that have come out between these. And what you can see from going from 2016 to where it is now is that we have a tremendous amount of interaction between those institutions that we believe might not otherwise have happened. And that is in part one of the major goals of the Biohub, is to realize synergies between these institutions. Each institution a powerhouse in its own right, But the truth of it is that there's a lot of barriers to doing collaborations between those great institutions. And the Biohub acts to remove the friction from the system to make those interactions happen more seamlessly and with ease, to make the science greater, faster, and better. Now, in addition to our investigator program, we have a lot of platforms. Platforms are like technology shops, if you will. So unlike core facilities, which are usually fee-for-service operations, like a flow cytometry core or something like that, these platforms are meant to develop technology and bring new things to the academic arena. So we have a bioengineering platform. I'll have more to say about that, but that's like electrical engineering, mechanical engineering, fluidic engineering, and so on. A data science platform, one of the largest, and it's the largest platform in a growing area at the Biohub, machine learning, deep learning, AI forward type technologies, and really making those and bringing those to use on data. Sequencing, genomics, always near and dear to my heart. And of course, we're developing new methods in that area as well. Computational microscopy, I'll have more to say on this. Mass spectrometry, the flip side of genomics, we need to know about proteomics and metabolomics, and so we're developing a lot of new tools and technologies there. And a community access program at Stanford called the BioCat. All right, here I'll just give a couple examples and then tell you a bit more about infectious disease. So the bioengineering platform, the idea is to make devices that serve the mission of the Biohub and its collaborators and investigators, and to bring them out in almost a product sort of way, but not sell things. They're all open source. They're all published and easy to do. So Rafael Gomez is the director of the bioengineering. There's some of his team members below. Shown up here is a portable luminometer, which is we use for field serology. So many of our global partners, like in Bangladesh and or other countries, would like to know how much of their population's really been exposed to SARS. So those kind of serological assays really generally require sophisticated plate readers or laboratory equipment, not stuff you're going to haul out to the field very easily. So we developed a handheld luminometer. It's very low power, can run for a couple weeks on batteries, and it has the sensitivity of a commercial plate reader. And we can get that out to places that need it. Actually, we have deployed to Bangladesh. They're using it in Dhaka. That's shown in the lab, testing it. And Shajudi Saha there is using it to assess the exposure of the general population around Dhaka to SARS, which nobody's been recording, like how many infections have occurred in that place. Another example, I'm giving you some examples from the sort of global health arena. This is something I really care about. Another example of a cool instrument from bioengineering is, again, another open source instrument for low-cost, label-free detection of blood parasites. For those in the malaria field, the 100-year-old technology that is used to diagnose malaria is to smear blood on a glass slide, state it with GIMSA, and then look through with your eyeballs and count them up one at a time. And it's extremely painful and annoying. And we do it in the lab all the time. And if you're out in the field, first of all, it's slow, it's prone to error, people's eyes get tired, and so on. So we've built a low-cost microscope that flows red blood cells by. I'll flip it and show the movie one more time. And as red blood cells flow by, unfixed, label-free, through machine vision, there's basically a neural net that's been trained to see what parasites look like. Those are the red ones that go by, you'll see occasionally. And we've got a paper in PLOS Computational Biology on this. It's way more accurate than the human eye. It doesn't get tired after counting 1 million cells. It just goes and goes and goes and doesn't require fixing or chemicals or any of this stuff. And so this is something we're looking to deploy in a number of different countries soon, and hopefully get some data back. And the more parasites it sees, the smarter the model gets. It's sort of like self-driving cars. The more miles that are driven, the better you can retrain those neural nets and get it more and more precise. And so this is something really fun. Now, on the computational microscopy side, the idea is we need better ways of imaging cells. We need better ways of getting data out of cellular systems and new kinds of microscopes that will give us higher spatiotemporal resolution. An example of that is from Loic Reuter and Bing Yang. This is called the Doxyscope. It's an oblique plane microscope. That means it uses a light sheet that comes at an angle out of the main objective, and then the image is formed back there on the right. And what that means is you can get a much bigger spatiotemporal resolution in single cells looking at organisms for long periods of time. And it's sort of auto-focusing and auto-calibrating as an organism moves around. It can move with it. The scope itself looks really ugly. but that's what breadboards look like. But this is what the data looks like. So the tail is over there. You can see some somites. And what I think you can, so this is sped up, obviously, so you can see it a little bit better. But you can see how we can identify every single cell uniquely through, again, through neural nets. And so knowing that, we can rewind this, and we can literally track the trajectory of every single cell. So now this is showing cell tracks. And of course, you can follow all those cells at any point in development in a long developmental trajectory. So as we zoom in here, you can see where all the cells are going. And why is that important? We're trying to figure out how our body's built. What is the coordinated action during development that allows this whole organism to be built? And looking at the coordinated movement and action of all these cells, combined with the genomics and the proteomics and everything on top of it, allows us to decode that program of how an organism like a zebrafish is built. And it's tools like this that enable asking some of those questions. And of course, again, all open source. It just got published, and anyone can build these with those plans. I didn't say it was going to be easy to build one. It's a little complicated, but they are pretty cool. Now, making images is nothing without the right software. And one of the things that I've always been a big proponent of is if you make a tool, you make like a software tool, get it out there. Make sure other people can use it such that other people can do the science too. And so image viewing in biology is kind of a big deal. Everybody's looking at microscope images. And the image viewers that are out there currently from the old days, like Fiji and NIH Image, they're not very good. They're really old. They need being updated. And so the Biohub launched a project called Napari. Actually, Loic Royer did it once again. Launched a project called Napari, which is a free open source Python-based image viewer for n-dimensional data, meaning time series data, 3D data, all different kinds of stuff. And it's really exploded online. We have over 140 plugins now and 300 developers developing plugins for this open source system. You can download it, get the GitHub repo, and it's super high performance. It's designed for GPUs. It's free, it's fast, and it can really do just incredible kinds of analysis. And if you're a developer, write a plugin for it and contribute back to the community. That's sort of the attitude of Napari. And so I think Napari really illustrates one of the themes of the Biohub, that we tried to get these tools and then push them out to the community in a way that's going to help people. All right, so on the genomic side, one of the biggest things that's come out of the Biohub is a ton of single-cell sequencing data. And that single-cell sequencing data has come in the form largely of these tabular projects, which are basically cell atlases of different organisms, trying to identify all the different cell types in both health and disease. Tabula miris, the mouse atlas, was one of the first ones. There have been follow-up tabula mirises having to do with basically during aging. The fruit fly atlas, the zebrafish one is coming. There's a lemur atlas and a human atlas, of course. And what this is all equated to is a lot of different cells, all sequenced at very high resolution. We've sequenced over 1.5 million libraries at the Biohub to get this project done, with tons of annotations. The data is all freely available. You can download it anytime. A lot of papers associated with it. And there's some really cool tools online. For example, cell by gene, spelled cell X gene, that allows you to then query that data, even if you don't have the programming tools, to answer the question, hey, where's my gene expressed? Which tissues, where, and when? And all that's in there. And so this cell atlas data is a really useful foundational data set by which you can then answer or ask questions or hypotheses with regard to your own data. Along that same line is OpenCell. So OpenCell is a really fantastic project led by Manuel Leonetti in the Biohub. And the whole idea here is to really figure out where all those proteins are expressed in the human cell. So he's been systematically, endogenously tagging every human protein, put a little fluorescent tag on it to understand under its own native promoter where are these proteins expressed, how much are they expressed, and who do they talk to using mass spec. And so this is a screenshot from the web page. You can get images and movies and Z-stacks for every protein that's localized, and all the mass spec interactions of who they talk to. And then you can just pick your favorite proteins and go in there. So currently there's about 1,300 proteins already tagged with images available, with mass spec data available. All, again, can be downloaded. You can use it for the basis for machine learning or other things. And it's all at OpenCell, czbiohub.org. And of course, the idea is to scale this to the other 19,000 or so genes that are in the proteome. And that's going to require a lot more robotics, but that's one of the goals we have. All right, now all this stuff is on itself fun, but we're putting it all together in new ways. One of it is a new project that's led by Sandy Schmidt, our CSO, called the infected cell. And it really brings different components from infectious disease like CRISPR-based screening and genetics, the metabolomics, the proteomics, as well as a lot of the lab automation and computational microscopy to compare and contrast how viruses subvert the host cell and what mechanisms they use and how they remodel the architecture of the cell, again, to fit their own needs. And knowing that, of course, we want to create interventions, therapeutics, or even tools to figure out how to study them and stop them from doing that sort of thing. Okay, I promise we're getting to COVID. Okay, so this infectious disease will take us right there. So one of the other initiatives at the Biohub that we started right when we began in 2016 was the Rapid Response Group at the Biohub. And they're a group led by Christina Tato to tasked with responding to things like outbreaks, surveillance missions, as well as preparedness for pandemics like tools, standards, training, various different pipelines for compute, as well as understanding things like vectors in different reservoirs like mosquitoes and population. And how does this actually sort of manifest? Well, the whole idea was, and this is pre-COVID, the whole idea was how can we put together the building blocks to create a global pathogen detection network? Now at the time, I said that, I would say, hey, how about we build a global pathogen detection network? And people are like, what's that useful for? All right. No one's saying that anymore. But at the time, we got a lot of blowback. And so how does it work? How have we conceptualized it? So the idea is to engage basically with partners around the world, especially in low and middle income countries that bear the highest burden of infectious disease. Through outreach and training, we do metagenomics or genomic technologies to understand infectious disease. That's our core strength of technology. We want to impart that and empower groups to build on that, and realizing, frankly, that sequencing of stuff has gotten super, super cheap, but the computation has not to solve that problem, that roadblock. That, so what if you can make a bunch of sequencing data? It's not useful unless you can analyze it. And so, again, through open source software pipelines that allow groups all around the world to get real-time data. And that package actually manifests itself as a program called CZID. It's on it's at czid.org. It's for real-time, super fast analysis of metagenomic sequencing data from literally anything. It could be from mosquitoes or humans or different animals. And it's meant to classify what's in that sample. Is it infected with a virus? Does it have a parasite? Does it have a fungal infection? What's going on? And again, it overcomes that barrier that while you can have a sequencer in the middle of Bangladesh or Cambodia, you often can't analyze the data. The compute isn't there to finish the job. And so by putting it in the cloud and making it freely accessible, we can solve and cross that barrier. And so putting those pieces together, we then teamed up with the Bill and Melinda Gates Foundation in a Grand Challenges grants program to fund groups for equipment, travel, and staff to provide that on-site training, the real-time analysis and data storage provided through CZI and the Biohub, and to effectively train people in countries so they become the trainers, train the trainers, and to ultimately engineer ourselves out of the loop. Our first place that we started working in was in Dhaka, Bangladesh. This is a picture from Dhaka Sishu Hospital. I work with Sajudi Saha there. And we really launched this program as a pilot to see what it would be used for. And her focus was on childhood meningitis. There's a lot of childhood meningitis in Dhaka, and over 50% was of completely unknown cause. Just no idea what it is. And that is, and they see not just like 1,000 admissions, they see tens of thousands of admissions per year in Dhaka with childhood meningitis. And so right off the bat, she's a microbiologist. She suspected there was going to be a lot of bacterial infection that they weren't seeing. Is that true or not? So using cerebral spinal fluid from children in Dhaka, she used the system. She did the sequencing in her own lab in Dhaka, put it up on the pipeline. And here's just an example that we saw right off the bat. Here's a 5-year-old with meningitis. She did not have a bacterial infection. In fact, in this case, it was chikungunya virus. a virus, a mosquito-borne virus, that most people did not appreciate could be neuroinvasive, and especially neuroinvasive in neonates. And that was completely off their radar. They had never considered it. And when they retrospectively went back and looked at a bunch of prior CSF samples from the years prior, they found lots of chikungunya, corresponding to when chikungunya was in country. And so, again, people tend to look for things that they expect. They're biased by their preconceived notions of what they think might be in a sample. Metagenomics, that is the sequencing of all the RNA and DNA in a sample, doesn't care about your preconceptions. It just asks, well, what's human, what's not human? And we'll figure out what the not human stuff is. And it gives you an answer back, whether you like that answer or not in some cases. And most people don't like the answer. But it's important to know the truth. All right, so where does this go? What have we been doing with it? Well, this whole system provides agile tools. These tools can be obviously used for pathogen identification and detection, but also for disease epidemiology. We'll see some more of that coming up. And vector surveillance and zoonoses, like the monitoring of the bats shown on the right from Madagascar. And of course, it can be used for environmental sampling, livestock sampling, and so on. So where are we today? We're in these countries today, right now. And pre-COVID, we're in about 10 of these, where we have sites where people are already sequencing, already looking at their local problems. We're not dictating what problems they look at. It might be childhood meningitis in Bangladesh. It might be mosquito-borne illness in Cambodia, or antimicrobial resistance in South Africa. But through the same system, all that data can flow through one cloud-based system, so you can basically see global real-time world analysis of different species and different human samples or non-human samples being analyzed. So that brings us to January 2020. Where was I in January 2020? I mean, everybody knows where they were probably in January 2020, right before the pandemic hit. I was in Phnom Penh, Cambodia, and we were setting up one of these detection nodes, training the staff there to use metagenomic sequencing and getting them online in Cambodia. And little did we know, like I left on January 10th and realized coming through the airport in China that something was really off. It was not the same kind of airport that I came through on. It was a very different situation. And literally just a few days after I left, Cambodia received its first tourist from China with COVID. And the system picked it up. Not only did the system pick it up, and this is, by the way, in the collaboration with the Pasteur Institute there in Phnom Penh, not only did our team pick it up, but it wasn't in the database. It was identified as bat-like SARS coronavirus, which is exactly what it is. And the full-length genome got sequenced on the system, even though there weren't fancy like primer sets or any of that stuff out. Metagenomics doesn't need primer sets, it can get the whole thing. And so it was one of the first full-length genomes deposited publicly from outside of China very early in the pandemic. When I got back to UCSF in San Francisco, the whole game had changed, and our priorities rapidly changed too. What can we do to be involved? How are we going to take part in this? We're an infectious disease group. We call ourselves Rapid Response. Is this not the Super Bowl? Like, we better do something. Are we going to do surveillance of asymptomatics? The CDC didn't want us to do that. Are we going to do drug development, vaccine development? What kind of basic science can do? And of course, what about clinical testing? Well, that became a really big issue because towards the end of January and early February, like many places around California, there was really a dearth of clinical testing. The big labs, Quest, LabCorp and stuff, were not returning tests anytime soon. You send it to the CDC, it might never, ever come back. And so, we needed to address this from our own local hospital perspective, but also from our community perspective, because we had a feeling, it turns out to be true, that a lot of our vulnerable populations in the Bay Area, whether they be people in jails or prisons or skilled nursing facilities or safety net clinics, would be uniquely vulnerable to this and the least likely to get testing or have access to testing early in the pandemic. So this was March 19th, 2020. This is my lab manager, Jen. That's a big empty lab at the Biohub. And we thought, okay, we have an empty lab. Right before this, on March 12th, there was an executive order by Gavin Newsom, the governor, that basically paved the way for us to get into the clinical testing game. Because of clinical testing regulations called CLIA, normally you have to have a certain kind of personnel in your lab that has certain certificates of training that require for laboratory testing? Well, I've got a whole bunch of PhD graduate students and stuff and postdocs that are super well qualified, but they don't have that certificate. So normally they wouldn't be able to do clinical testing. The executive order by Gavin Newsom allowed this to happen with trained individuals not necessarily possessing that certificate. So it was game on. March 13th, empty lab. The students got together, the laboratory Biohub folks, and said, well, mapped it out. Could we do this? What would it look like? And that grease board is literally like the command center. That's Vita sort of explaining how we're going to do it, and it's going to be a PCR test. And then what we did is basically asked all the people at the Biohub to stop what they're doing. You're not going to stop the zebrafish for a minute. So everybody got assigned to different teams. Our lab leads are Emily Crawford and Vita Ong. And we had our CLIA lab directors we were able to borrow from UCSF, Ed Thornborough, Steve Miller in particular. And so with a team of different individuals, we were able to divide it into different groups. Everybody got assigned to teams, some of them not glorious, like waste, some of them more glorious, like the RT-PCR. And we were able to put these people on teams, each with team leads and second in command. So if somebody got sick, the other could like fit in, and we had non-overlapping squads coming in just in case one had an infection and took the whole squad down like others would. It was a thing. A lot of planning went involved. Now, one of the things that I did do that I actually, a lot of mistakes were made, but one of the things that I did not make a mistake in is right when I got back from China, that my first stop was not to the RT-PCR station or anything like that. When I got back from China from the Cambodia trip, I went right into our stock room. And I wanted to know, where does all this stuff come from? Does it come from overseas? Where does our stuff come from? Like pipette tips and chemicals. And we basically made a big inventory of where all our inventory came from. Goritza, our lab manager, went through a list and said, here's the stuff we import, here's the stuff. And I thought, okay, look, if this gets bad, there's likely to be supply chain disruptions. What is the stuff we can source locally in California? What is the stuff that we can't source from California? And in the next 2 weeks, I want you to buy everything you can. And so we stocked up. I spent millions of dollars stocking up my stock room. People thought I was crazy, but turned out to not be crazy. Now, if you all had trouble getting pipette tips at UCLA, I'm super sorry. You know where they went, and they went to a good use, though. I know Oliver's looking at me like, maybe that wasn't such a great thing. And what looked silly at the time wasn't silly later. Now, I'll give you a little timeline of events. March 11th was when that executive order went out. We started on March 12th. And by March 20th, 8 days later, we returned our first CLIA-approved clinical test from a patient. 8 days to build the entire lab. And I had 175 volunteers. It was intense. Because all those graduate students and postdocs sitting at home on lockdown, they wanted something to do. So, do you want to be part of a CLIA lab? Game on. It was no trouble getting people. I put an email out, and I was flooded with responses. And so we had tons of people show up. And of course, like every good graduate student operation, we fed them meals. As long as there's free food, the graduate students are going to show up. Maybe they even showed up just for the food, actually. But I like to think it's for the testing. All right, so where did we get to? We ramped up to about 2,680 a day, which today doesn't sound that much, but in April or March of 2020, that was actually a lot. And it's a pretty straightforward task, like the samples come in and they get processed for PCR. We ended up extending our services to all the counties of California. Now, not all the counties took advantage of it, but specifically the departments of public health. So many of the DPHs in the various counties did not have very many resources. It's very uneven how the different DPHs are throughout the counties of California. And so we wanted to extend free, like no one pays a bill, we're not billing insurance or anything, free testing to jails, prisons, DPHs, safety net clinics, and other vulnerable populations. And we had about 24 or so of the counties take us up on that offer, and we did routine clinical testing for many of these hospitals and counties, and some of the top users are shown up there. So I'm just going to take a moment and just take you through like life at the CLIA Hub in March 2020. Random people showing up in parking lots, dropping tubes off from counties all over. People would drive down from Lassen or Modoc County and just drop us off at the parking lot. Tubes have to be accessioned. We couldn't enforce a normal accessioning system, like make people use barcodes and stuff. So we had teams of graduate students and postdocs, including myself, hand typing in names and stuff, because there was no way for them to use electronic system in many cases. We couldn't enforce that and still do testing. And then all these accessioned tubes then go in and get transferred into plates. We did it for many hospitals as well. And from plates, then RT-PCR, and then resulting out. So we try to do everything within 24 hours at the time. But this accessioning business, like actually typing in like a prisoner's name and cell block and all that by hand, this is what a lot of they're doing, is going through paperwork and all this, was incredibly taxing, and it actually took the majority of the effort because no electronic systems were in place. Oftentimes, they couldn't even fax us anything. Ugh. Fax machines. Okay, so this was kind of the state of the affairs. Now, one of the things that we ended up doing with the CLIA Hub was actually putting it to work, not just for these different DPHs, whatever samples they wanted to unload on us, but also in our own backyard. We needed to use this power for our own populations in our own neighborhoods. And so this was, I'm going to tell you about a collaboration between the Biohub and Unison Salud and the Latino Task Force at UCSF, also in partnership with SFDPH, CalDPH, the City of San Francisco, and many others. And my partner in crime on this is Diane Havlier. She's an HIV doc and researcher at UCSF, but really turned her full force of attention to this, together with Karina Marquez, Maya Pearson at Berkeley, Diane Jones at UCSF, and community organizers like Susana Rojas, who is part of the LTF. And the idea was, you know, in April, this is March or April 2020, what is going on in our neighborhoods with COVID? Who actually has COVID? Because the impression we had is, there's COVID somewhere in the city, but where is it? Nobody really knows. It was like for testing, the idea was, well, if it was like a fire, let's just drive fire trucks randomly around the city and squirt water here and there. Wouldn't you want to go to where the smoke is? But you don't know where the smoke is unless you go look. So the first thing that we did is we took a hard look at our Mission neighborhood in San Francisco, which is one of the most densely packed neighborhoods, to do literally door-to-door free testing, community testing, asymptomatic or not. Remember, at this time, many organizations were discouraged from testing asymptomatic people. We now know that is completely backwards. But at the time, we were being discouraged from doing that. And of course, we'd give back to the community in terms of regular updates on COVID, rapid data analysis and distribution, and a lot of follow-up. And we were told this wasn't going to work. A lot of reasons why we couldn't do this. There's no place to do the testing. There's no protocols to handle the sick patients. People are unwilling to test if they're asymptomatic. Is testing even safe? Who are you giving the information to? Are you giving the information to ICE or the government or something like this? A lot of our individuals might be undocumented. You don't have any systems to store the data. You don't have any labs to perform testing at scale. So obviously the Biohub solved some of those problems, and with the community, we solved many others. So putting all the pieces together, we have working with the community, the LTF, we have that intervention development, the community mobilization. If you don't have trust in community mobilization, no one's going to take advantage of services you offer. The science through UCSF and the Biohub, the service, so the data information systems to get information and data back to people quickly, like the comms, the apps on phones, all that stuff. And of course, you have to have government partners. And so partnering with SFDPH, the city, and CalDPH was critical so that we're communicating clearly on guidelines and what we're doing and that everybody's on the same page. So in late April, we launched a 4x4 block area to get everybody within that 4x4 block area. And that's about 4,000 adults in a 4-block area of the Mission door-to-door testing. It's about 58% Latinx and about 34% below 50K household income. And so it's a very diverse, very interesting place in San Francisco. And many of you know these results, but this was the first big eye-opener to us. Now, we did serology and PCR. Let's focus on the PCR. That is, who's got COVID now? 96% Latino or Latinx, and 4% Asian. And even though there's over 1,000 Caucasians that lived in that neighborhood, there was essentially nobody in that population with COVID. So when we say inequality, like totally bizarre distribution, this is crazy. And so it really opened our eyes. This was a population that was being or bearing an unusually high load of COVID, much, much more different. In other words, you have people in the same neighborhood, in the same apartment building, but their risk factors are not the same. If you're going out on a contracting site or in the food and beverage industry or something like that, you don't have the privilege of being on Zoom, the privilege of being on Zoom all day, the privilege of being on Zoom all day, crushing some code, for your tech company. You are outside working, and so your risk factors are not the same, even though you live in exactly the same place. That was made blatant by these door-to-door testing things that we did, and refocused our energies to figure out what was going on. The other big wake-up call for us was in April. Obviously, we had data from the Diamond Princess that suggested it, but in our data, like when you have your own data, like you really get it, 53% of all the PCR positive people never had symptoms. And on 2-week follow-up, never did have symptoms. This was the sort of moment that sort of, the abyss opened up in front of me mentally, because I thought this was going to be like SARS-1. Everybody who got SARS-1 got symptoms and could be quarantined and controlled, and it was like died out pretty fast. If half the people don't have symptoms, how can we possibly control this? You won't know who has COVID. And that's exactly, unfortunately, what's happened. It's very difficult to control the virus for which a large proportion will not have symptoms because you just don't know. All right, the other thing that we realized early on is that we needed to use genomic epidemiology. And so I have to say a little bit about that. What is genomic epidemiology? So in addition to testing people, knowing they're positive, we also know what the sequence is. And what can the sequence tell you? Well, imagine like hypothetical county like that. I'm not going to go too close to the screen. I learned my lesson. You got random individuals around the county. Some of them work in factories, some of them work in a school, or maybe some of them work at a skilled nursing facility. We can tell from the COVID sequences a lot because the chain of mutations is somewhat unique. Mutations are introduced essentially randomly at a basis of a new mutation every 2 to 3 transmission events. So it's like a breadcrumb trail, which you can track where COVID has been and who it's been through, and then change a map like this to a map like this, where, okay, all those random people that were positive, I now know that 3 of those got it from each other at the factory, but not the 4th person. He was an unrelated infection. There's a new mutation at one skilled nursing facility that's causing an outbreak. There was people linked between 2 facilities that we didn't know were linked because they had undisclosed second jobs. All of that stuff, by the way, is real stuff. And this provides sort of a tactical intelligence through the sequencing that we otherwise wouldn't have had. And so we launched a program called Covet Tracker. It was a statewide collaboration in collaboration with CDPH, and our partner in crime there initially was Charity Dean. And the idea was to generate the data from the departments of public health, the public labs, all the samples that we're sequencing. We would build the genomes and phylogenetic trees and provide visualizations to our DPH partners who don't have time to do that kind of informatics or don't even have the people to do that informatics, so that we can conduct hands-on sessions to help them create interventions. For example, 5 people got COVID at a fruit packing plant, and 3 of them are on the same packing line. Did they get it from each other, or did they coincidentally bring it in? If they got it from each other, maybe the health, the safety rules inside that factory need to be revised and needs to be temporarily shut down. If they coincidentally brought it in, the factory is not at fault here. So it's a rule in, rule out. And so we did dozens and dozens of those interventions with the state, working on them to change the sequence into public health action and policy. And that was extremely effective. If we use that technology, just genomic epidemiology, on the mission data that I showed you, that 4x4 block area, what does it look like? It looks something like this. Focus on the diagram at the right. The x-axis is the number of mutations. So you can think of that as time. And each individual on here is someone who has who's had COVID. And for example, the yellow, I'm not going to get too close to the screen. I'm nervous because the sound thing's going to happen. But that yellow orb there in the sort of the center with the 3 blue dots above it, that's someone who's antibody positive for COVID, meaning they had it. And their SARS is directly linked to 3 people right up there who don't have the antibody yet, but are positive, and they all live together. And they all have exactly the same one. But they're also linked to those blue triangles, 3 workers that all live together, not in the mission, but they work in the mission. And they're all linked to a 4th individual, another blue circle down there, of a resident in the mission. So you can see introduction into the mission, out of the mission, back into the mission, and and then you can track all these infection trees down the line. And by these little yellow arrows on the left, we knew that it wasn't just one continuous outbreak in the mission. It was several introductions coming in over and over again, generally with short transmission tree chains outside of that. Now, if all things Biohub, there's going to be some software behind this, and there's more. So this is CZ Gen Epi. It's a whole workbench for genomic epidemiology for public health officials to be able to use genomic information to create actionable intervention. And that's available at czgenepi.org. I'm not going to spend time on it, but there's a lot of stuff you do by hand. This can automate a lot of that stuff. All right, so after doing that 4x4 block area, we said, okay, we need a presence in the mission, and it needs to be ongoing, everyday testing. How is it going to be most effective? Put it at the subway station. Put testing at the subway station. So at the BART, if you know San Francisco, the BART system is our subway at Mission and 24th, really at the heart of the population map. And so, again, in collaboration with those same entities, we set up testing there. Low barrier, no appointment necessary, no phone app necessary, no documentation necessary, no ID necessary. Just stand up and get tested. As well as when vaccines came along, more vaccination and other services too. We'll have more to say about that. One of the criticisms we had is, well, you're only going to get people that live around the BART station, like actually at BART, at 24th and Mission. Is this really going to have any reach beyond that little locale? So if we took just a few weeks of testing and map it onto the map, this is where we got to reach. Throughout the entire city, and in fact, 8 counties. People would come from all over the Bay Area to go to this testing station because it was super easy and didn't require appointment and didn't require a car and didn't require an app or any of this stuff. And so we had tremendous reach. Now, if you live in San Francisco, you'll observe that sort of red portion that kind of snakes through. That is the BART line. So obviously, being close to a BART line allows you to get to the testing easier. That goes without saying. And many of those other areas around the Bay are BART stops as well. And so extremely successful at getting a good population distribution from the city at a single site in the city. This is not a huge site, it's a little site. So we began doing additional campaigns to understand what was going on in there. So this was sort of getting into the late spring, early summer of 2020. And we did a snapshot of just, okay, what's going on right now? We did about 2,600 people in just a couple days, 9% positivity at that time. And once again, 93% PCR positive for people of Latinx origin, meaning their risk was much higher than whites or Caucasians tested in the same place. And you see some of the breakdowns down there. 23% unemployed, 16% food and beverage, and so forth. But the key thing was 87% were low income and lived in large households. In fact, you can get the same distribution, not by asking if you're Latino or Latinx, but by, do you live in a large household, more than 6 people? Do you make less than $50,000, and do you work outside the house? If all 3 of those are true, you use the same risk profile, much higher risk of getting COVID. So we then realized, and this is really a huge kudos to Diane and her team, realizing that testing alone is not enough. What you do in the laboratory, it can't stop there. And so Diane and her team really designed this test-to-care model. And this test-to-care model is really kind of a 360 support system. So besides getting the clinical test and the clinical assessment, lining it up with social work support as needed, ongoing assessments for 2 weeks, delivery of food and income replacement to the house. If you're the sole breadwinner of a house and you tested COVID positive, are you not going to go to work just because you have COVID? Of course you're going to go to work. You're the only one making money in the household. So you needed income replacement, and we needed legislation passed in San Francisco to prevent you from being fired from your job for quarantining, which was still a thing. It was not illegal to let someone go. And so this test to care model was super important, as well as figuring out if they had primary care physicians and so on. So this full 360 care really was meant to break transmission chains. Let's get them food coupons and deliveries so they don't go to the store, so they don't get out of quarantine. And that was really successful. But these community projects are also living laboratories that allow us to ask super fast questions because we have all the clinical metadata to be able to answer these questions. So for example, early on in the pandemic, we were getting queries like, I heard children are super spreaders. They have high viral loads. I heard they're super spreaders, and I don't want to be like around kids. Okay, I can get the not around kids thing. I've got teenagers, and it can be difficult sometimes. But not because they have a higher load. And there was a JAMA paper that came out, and here it's from Healthline back in 2020. The data showed they have a higher viral load. Well, because we have the living community laboratory, and we get all age ranges from like 1 to 100, I can just go ask that question without a moment's hesitation. So we did. There's the viral load distribution on the y-axis for the age decades along the x-axis, and there's no statistical difference from 1 to 10 to 91 to 100. It doesn't matter. So good news, kids aren't viral super spreaders. Bad news, same viral load as everybody else. I mean, they're not less either, in other words. And so these are the kind of questions that with like one database query, we can just answer, and not with like 15 or 20 kids, as some papers published, but with a couple thousand people really fast. And this was early in the pandemic. Obviously, all those systems are up to date, and most people can do that now. But at the time, there was a dearth of information. People would ask a question, but nobody had the data, or if they had the data, they weren't showing you for some reason. Now, the other thing that we came to the conclusion on in 2020 is that turning a test around in 24 hours was not fast enough, not acceptable, no longer okay. Someone gets a test, they don't know the answer till tomorrow or the next day, they go back to work or to their job. Like, oh, but maybe they're positive. How do you know? And so, this is a graphic from Michael Mina. It's now famous, sort of like a kinetics of the viral load as per unit time. And there's this critical area where people have the most virus and the most infectious. And there's this long tail where we probably don't really care about that much because they're not really infectious anymore. And so how do we get quick answers? And this is about the time that direct antigen testing came along. And so one of the first products was Binax Now, COVID antigen test. They detect the protein of the virus, not the nucleic acid. About 15 minutes. They're about they're not $5 anymore apparently, but they were about $5. But they weren't available. At that time, you couldn't buy them at Walgreens or anything. They were not over the counter. But it doesn't require lab equipment. It's simple deploy. It's super fast. But do they work? We had no idea. So CalDPH got us a bunch of the tests, asked us to evaluate them in the context of the mission project. First thing I did is took it to lab. We grow COVID in lab in a BSL-3 so we could test real virus on real cards and calibrate it for how it did against PCR as well as viral culture. And those results were really encouraging, that down to a cycle of threshold of around 30 or even beyond, we could see virus on the card. So this looked like a plausible way forward for us to do cheaper, better, faster testing. I want results in 15 minutes, not next day. And so we then implemented it at the mission. So here's a whole bunch of people, 3,298 that we did comparison PCR versus antigen test with the general population. Just walk up at BART. And the blue is where PCR and Binax are in concordance, and yellow is where they're not. So at the very high end, with very tiny viral loads, you're right, it's not as sensitive as PCR. But I would argue I don't care. That's the region where people are less likely to be infectious or not infectious at all. And we really want to catch this big middle ground where people are the most infectious. Now we can put real statistics on that. And so what we figured out is that even if you don't use a cutoff, the sensitivity and specificity was really good. If you compare it to PCR to like a cycle threshold of 35, we're looking at 99.5% sensitivity and 99.6% specificity, meaning very few false positives. That in my book, like one false positive per 1,000 at best. That's okay. I'm willing to take that. So we rolled with it. And the impacts of these studies up through December was, again, We established those neighborhood sites. We had new testing criteria. We expanded the community test-to-care model. The wage replacement, right to recover, was introduced by Supervisor Hillary Ronen and was passed in San Francisco. That provided income replacement to people who needed to quarantine. And all that data we generated for the FDA, we were able to just throw over the fence and was used in the FDA approval for Binax Now for over-the-counter. So right after that, it was able to be, people could just buy them at Walgreens, assuming they had them in stock, which was a problem. And so that was a big impact. Now, at the same time we were doing all that sequencing, we were seeing these changes. And by January, we saw that different variants were beginning to take over. This one is called the epsilon variant now. Back then, it didn't have a name, but it's characterized by this mutation at 452 in spike. And we could visualize that here. Here's a circular phylogram like next strain showing the growth of this yellow and red strains that were just taking over in the mission that had this new mutation. But the question was, early on, actually this variant was first detected in LA, the very first one. And then it really came up to San Francisco. And there was a kind of a furor over it. This was the first variant that was really taking over. Everything else was kind of equal opportunity. This one's really busting down. And the question was, well, how bad is it? Is this really as pathogenic? So the way you answer that is with a community model again, because we can measure a bunch of different things. First of all, no difference in viral load. Second of all, same percent of people with the symptoms. No difference in hospitalization. So right off the bat, everybody should just calm down. But what about transmission? It is taking over. We can do what's called the secondary household attack rate. This is better than a laboratory because it measures when you put an infected person in a house, how many other people get sick. That is an operational measure of how transmissible the virus is. And what we found is, whereas all the other variants were about 26% secondary household attack rate, meaning a quarter of people get sick, 36% for this new variant. So elevated, it's higher, but it's not like so crazy high that we need to hit the panic button. But it was sort of a harbinger of things to come, that's for sure. All right, and so additionally, because we have super deep data, and I'm getting towards the end here, so bear with me. Am I good on time, Tracy? I'm good. All right, that's what I want to hear. Is that we can really drill down into the data. And we were beginning to see, as vaccines rolled out in 2021, that there were breakthroughs. We could see people who were vaccinated already getting sick. And so we had a case of a household. This is one of the first breakthrough households we looked at. And a bunch of people, there's the P1 strain. The diagram is complicated, but the index case is this 80-year-old male in the middle, Z8. And they had the Pfizer vaccine, and a bunch of people in the house. Some got it, some didn't. What was behind all this, and what's going on? Well, we could look, because, again, it's a community project, and we have community buy-in, we can ask all these people to come back and give us blood samples. So we can look at their antibodies and see what's going on. Looking at so-called neutralization. That's what this curve is. The gray curves are sort of where the normal population is in terms of neutralization. Here's individual Z8 in the house. Toward the right means less neutralization, not as good at blocking SARS. And this person is below the population average, like not great at neutralizing SARS. Maybe that's how they got it. Here's the other person in his house who did get it in the normal range. So even though they're in the normal range, they still got it. Here's another individual in the normal range, and they still got it. So what this is showing is that while the vaccine definitely protects, less people get it, and it definitely keeps you out of the hospital, doesn't mean it's a guarantee. You can still get SARS even though you're vaccinated. Here's a person that quarantined separately. Good for them. They didn't get it. And then there's this individual that went shopping with these individuals like all the time and didn't get it, even though they were exposed heavily. That's their neutralization. I mean, they're like, there's a 60-year-old female that has got awesome antibodies. But I'll tell you what, vaccinated, but probably also, we believe, also had it. So got that special booster and never knew it probably, probably asymptomatically. Now what's up with Z8? What was the risk factor for that person to get it? Yes, they have less neutralization. Maybe they were just unlucky, but actually there's more to it than that. Right about the same time, it became realized that there were certain risk factors for getting SARS and severe disease. And one of those risk factors were autoantibodies to type 1 interferons. This is work that Jean-Roland Casanova originally published. And when we test individuals for antibodies to type 1 interferons, this person's off the chart. They're making tons of autoantibodies to their first line of defense. If you don't have interferons, you are going to be at risk for severe disease and getting it. And so this is the paper on autoantibodies from Jean Laurent's group. And so this is really interesting, but how common is this? I mean, it was published in one paper. I saw it once. Is it something more than this? So we looked in our populations. Our COVID-19 critical patients and severe patients, indeed, we found it. About 15 to 20% of our severe or critical patients had the antibody. In moderate or moderate critical, never found it. And in the general population, because we have the Mission Community Project, let's just go look at 4,000 individuals and see if they have the antibody. You know, we found it in very few, like 0.3%, which is consistent actually with the population averages. Others have found it at 2. So what's really interesting is that this is probably a pretty rare phenomenon that people have these antibodies. It is 0.3%, not very frequent. However, in a pandemic where everybody's getting infectious disease, where this makes you uniquely vulnerable, it surfaces this new vulnerability that no one even really appreciated knew existed. And I would argue that every ICU clinic should have, type 1 interferon autoantibodies at their front line. If you know they're positive, you already know they're the ones that are going to be intubated or on ECMO or, God forbid, don't make it. And when we think about other respiratory viruses, is it just SARS? What about influenza? What about RSV? What about parainfluenza 1, 2, 3, and 4? What about metapneumovirus and bocavirus? We know our older populations tend to be more vulnerable to these respiratory viruses and oftentimes with fatal consequences. How often are they afflicted with these antibodies? And we know that the occurrence now from work of Paul Bastard and Jean Laurent that these autoantibodies increase with age. Although how they exactly occur is not known. So again, community projects. So here we are in fall 2021, and the question is about Delta and vaccination. If you're vaccinated, do you have less viral load? Are you okay to go out and about if you're vaccinated even though you got Delta? No, definitely not. Even if you're vaccinated, your viral load's the same with Delta. And so again, we compared our results as well as with Davis, and there were no differences in viral load, whether the subject was vaccinated or not. And I think that was really interesting. And then here we're coming up to the end here. Then came Omicron. Omicron wiped out all the other lineages. It was an amazing thing to watch from an evolutionary virology perspective. But the questions were, and these are the practical questions we can answer with the kind of community project we have. Hey, does the rapid test still work? There's a lot of folks on Twitter saying rapid tests don't work for Omicron. Was that true? I mean, I don't know. And then there was Twitter people saying, you shouldn't swab your nose. You should put the swab in your throat. Okay, there's a lot that can go wrong there, And so is that true? I mean, I think it's a bad idea, but is it a bad idea? I can go to the mission, I can swab a bunch of throats, so we could go find out. And to what degree are you protected against, if you've had Delta, are you protected against Omicron? So on the throat thing, we won't spend much time on this. Don't swab your throat. It's a terrible place to swab your throat. We did a lot of them, and the nasal, the mid-nares is a superior place if you want to pick up COVID. If you want to miss half the infections, go ahead, swab your throat, and you'll probably throw up. So let's put that to bed. Okay, and the tests do work. I'm not even going to show you a slide on that. They work great. All right, so back to this. What about these other questions? Are you protected? What if I've had the vaccine or boosters, or I was exposed to this or that variant, for those who know? Am I protected against Omicron? Now, the way to do that, again, is with neutralization. And we make pseudovirus vectors, meaning we make a virus with a customized a spike protein from one of the variants, like Omicron or Delta, and then we can put it in a virus that has a green fluorescent protein. And the simple question is, we can add this virus with patient antibodies and then put them on cells. If the cell turns green, the antibodies didn't work. If the cells don't turn green, the antibody's working. It's neutralizing. And then we can actually use all the different variants that we made against different sera. And the beauty of the Mission Project is we've sequenced every single positive, even because we can sequence off the Binax cards too, by the way. That's how we get those. And we can since we sequence all the positives and ask them to come back after they had convalescent antibodies, I can have groups of individuals that had all the different variants. So all their serums are unique to the variant they had. It's not just mystery sera. And so we were able to do the all-by-all sort of comparison of variants versus sera. That's what this complicated map looks like here. And so on the side, on the left side, are the different variants, and on the top are the different sera collections from sequence-proven different genotypes. Red means doesn't neutralize really well. Blue means neutralizes pretty well. And so what you can see is, so P2 right here, that nice blue one in the middle, P2 has the 484 mutation. So people who had the 484 mutation, that neutralizes really well. In other words, you neutralize what your immune system has seen before. It trains on what it's seen before, and it does really well on that. But when new things come along, not so much. So look down there at Omicron. If you look at Delta, the last wave we had before Omicron, it's among the worst. So your protection against Omicron, if you had Delta, is not worth anything. Maybe a little bit. But this is like log base 2 scale here, and so it's really down. But look to the right on the vaccine doses plus previous infection. Not bad. And in fact, the 3 doses of vaccine, and by the way, I've had my 4th shot, on the far right, that does among the best. So this really shows that the vaccination boosters really do give you a boost up. Now it does wane with time, and there's all that issue about how long to the next booster and so on. And I do hope we're going to have second-generation vaccines that incorporate many of the variants that we see out in the wild today. But this answers the question about, well, to what extent am I protected against this variant or that variant? Depends on what exactly you had. Exactly what you had. Now, I'll finish with just a couple unpublished pieces of data right from the mission. CDC and others had recommendation that if you've had COVID or Omicron, well, you can come back to work after 5 days. Basically, I mean, they don't actually say that, but it's like 5 days recommendations when you come back. Is that true? How many people still have virus at 5 days? Where's the data? Well, that's what the Mission Community Project is. Since we can just ask people to come back, we test them every single day. And so this is what it looks like. At 5 days, 65% of people still had antigen-positive virus. And at 10 days, 28%. This is all 100% Omicron, by the way. And at 14 days, 5%. So that's not great. Basically, people need to either do testing, antigen testing at home or something to really know. Just because you've waited 5 days doesn't mean you're clear. You might be in the 40%, but you might not be. In fact, chances are you're not. Now, what if I stratify this curve by vaccination status? Like if I'm boosted, do I get a pass and I can come back at 4 days or 5 days? Is that because I've heard a lot on Twitter about like we should have different tiers depending on where you're vaccinated or something, because if you get it, maybe you can come back earlier. I'm not even going to ask. I was going to say if it hazarded any guesses, because it's not great news. It didn't matter. It didn't matter. So here's the deal. Vaccination does lower your chance of getting it in the first place. It definitely keeps you out of the hospital and prevents severe disease. No question about that. Once you are infected, your lot is the same as everybody else's, though. Your viral loads, which I showed before, are the same, and your clearance time kinetics are about the same. So you can imagine the antibodies almost as like a wall around the castle. And once the troops are over the wall and in the castle, different game. It's not the same game anymore. And so maybe that's a bad metaphor, but the point is that you should, regardless of vaccination status or whether you've had that infection or this, you can't treat yourself any differently. You should be quarantining for about the same time as everybody else. You don't get a pass. But also, get your vaccines and boosters. This should not be anything interpreted to say, oh, I shouldn't get a booster or anything because it doesn't matter. It definitely matters. Your chances of getting it in the first place are less. And long COVID is a thing. I'm not going to talk about long COVID, but it's real. And so is neuro-COVID, which is the worst version of long COVID. And whatever you can do to avoid infection is a good thing. All right, where are we today? This is the mission district sequencing from the very beginning all the way through. That yellow-green is the Delta wave. Epsilon is buried there in sort of the teal. And then the orange and the red is all Omicron. And now there's BA2 up there. We're at about 30% BA2. It's holding steady. We're not seeing a big bump yet. There probably will be a bump, but I don't expect an Omicron-like mega peak like we had before, because the protection from BA1 to BA2 actually should provide some immunological headwind because the mutations in RBD, the receptor binding domain, are almost identical for all intents and purposes. So I want to leave you with this. I think we should have more living laboratories. One of the things that I've learned is this tight partnership in the community, like we have with Diane Havler, Latino Task Force, and our neighborhoods, allow rapid questions to be asked. It allows goods and services and testing and other things to get to the community quickly, while also providing the basis with which to contribute to public health policy, pandemic response, and basic science. And you do this not in isolation in your lab, but by working with partners in the community, with the people who really are getting the disease. And so there's a ton of people behind this. There are hundreds of people behind the Latino Task Force, Unitas and Salud, and at the universities that contributed to making this happen. I called out many of their names during this, and I gave references to papers at the bottom. I'm not going to be able to say all their names, but a huge thank you to Diane Havler and Janae Pilarowski, Rob Nakamura of CDPH, James Peng, John Schramm, Karina, Maya, Diane, and Susanna Rojas. These are key individuals without which we couldn't have done this. We have, dozens and dozens of volunteers. Here's some that I could get the pictures for. And these are graduate students and postdocs that spent countless hours typing in data. They're not getting paid. They did get free food. And we're really made this possible where it couldn't have been possible otherwise. Every great operation's got to have a cool t-shirt, so do we. And there are many, many Biohub people to thank. And early in this, Emily Crawford did a huge amount of leadership, and so did Vida Ayong in getting this off the ground. So I want to acknowledge them. And with that, I'll quit.